University of Panama and also at the INDICASAT. And he's going to, to talk about molecular tool, to study, medical entomology, especially mosquito. Thank you, Jose.
Gracias, Yves. Good afternoon, everyone, and thanks, Yves, for giving me the opportunity to share with you guys some of the research that we've been doing at INDICASAT in the last couple of years. I work on mosquitoes for the most part, and I look at disease transmission, ecology of mosquitoes, but today I'm going to shift a little bit to ticks. And I'm not talking about metabarcoding of insects, but we have done some metabarcoding of bacteria on ticks, and I think this is relevant. But I'm going to showcase data on two other molecular techniques that I think are important, especially now that we have this heated debate about whether or not using morphology only or molecular tools only, but integrating them on all these techniques. So I'm based at INDICASAT. I'm the academic coordinator of the master program in entomology at the University of Panama and a research associate at STRI, as well as a member of the SNI. So Panama had been considered a hotspot for disease historically and currently as well. So I'm going to quickly share some important aspects of Panama, which I think are relevant for disease ecology. For those that are listening virtually, Panama is located in the narrowest point of lower Mesoamerica. We have a tropical climate. We have torrential rain pretty much the whole year, I would say, and hot temperatures. So these conditions are really beneficial for insects which can develop year-round in Panama. We have the canal from which we have a lot of merchandise and goods coming across, and a lot of people as well. And this is seen as a conduit for the introduction of pathogens, people that carry diseases, as well as arthropods, such as mosquitoes. Panama is the second most populated city in Central America, just after Guatemala. And we have this marked pattern of socioeconomic inequity, which means that certain group of people in neighborhoods of low socioeconomic status are more exposed to mosquito bites and the diseases that these mosquitoes transmit. We have significant deforestation going on across the country, and particularly in eastern Panama, close to Colombia. And this has been associated as well with the emergence of certain pathogens that cause disease in humans. And in the last 6 years, we have this migration crisis. People coming through the Darien Gap may pick up some pathogens along the way and transport them to the northern hemisphere, or could be introducing pathogens from abroad, from places such as Africa, for example. And so these conditions are like the one that people used to think about Panama as a hotspot of vector-borne diseases. Historically, Panama, and some of you know that when the workforce was trying to build a canal, especially the French government, there was a lot of losses due to things such as malaria and yellow fever. And so due to this, the institutions such as the Gorgas Memorial Laboratory and the Smithsonian Tropical Research Institute established a research agenda about tropical medicine in Panama. And Panama has a huge legacy of work done on vector-borne diseases. Nowadays, as you saw, we have a steady economic growth, at least post-pandemic, post-COVID-19 pandemic, but we still suffer from the same issues. And we even have new pathogens, emerging viruses such as chikungunya and Zika. And we have actually new vectors such as the Asian tiger mosquito, Aedes albopictus. And so we have done a lot of work on tropical diseases in Panama, and we have not been able to solve this issue sustainably. So we need to do more work on it. As you go through a gradient of habitat alteration, there's changes at the ecological and evolutionary level that facilitate the emergence and the spillover of some pathogens. We have, for example, that the wildlife animals get replaced by farm animals and domesticated animals. in rural areas, and then people are the main reservoir of these diseases in urban areas. We also see changes in the mosquito community. Most species of sylvatic mosquitoes disappear as we go across the gradient, but certain species are able to cope with the negative changes in the habitat, and they become more abundant as they adapt to live along with humans. And this is the case of Aedes aegypti, the main vector of dengue, for example. And so viruses have to adapt to new species along the gradient, and as a result of that, they become more pathogenic and more virulent. And so most of the work that I've been doing, and folks in general in Panama, have been focused on mosquitoes. But there are other group of animals that I think are important, especially in animals and humans as well. And these are, for example, ticks. And ticks are external parasites, which we call ectoparasites. And so they rely on blood fully, all the different life stages, larvae, nymphs, and adults, both male and female, feed upon blood. They're bloodsuckers, and they need that to be able to reproduce and to move and to molt into different stages. And so we have a phenomenon which I think people have already pointed out in this event, which is called cryptic speciation. And so the morphology is basically the same. They resemble, different species resemble each other quite similarly at the morphological level, but when you look at the genome, there's significant divergence at the molecular level. So these are separate evolutionary units, different species. And so this makes it really difficult to identify ticks because they have this ectoparasitic habit that kind of converge in shape, and all the species kind of have the same shape. We have also significant dimorphism, so between nymphs and between adults, both males and females, there are differences. So you have to develop taxonomic key based on morphology specifically for each of these stages. And so far, we have only developed these tools for adult ticks. We can only identify adult ticks at the species level using morphology, but it's really tricky to do this at the nymphal and larval stage. And this is really tricky when it comes to running epidemiological studies, because you have to identify hundreds, if not thousands, of individuals. And so we need complementary strategies to identify these creatures. Here's one of my students sampling ticks. This is really fun. In the forest, you drag this piece of cloth, and as you drag that through the grasses, nymphs and larvae kind of grab on, and you can collect them. This is an active way of collecting ticks. If you're about to collect adult ticks, some of them climb all the way to the tip to the tall grasses, and they spread the first pair of legs, and this behavior is called questing. And so you can wear a white pant, as Philip is doing, and walk through the forest and then be able to pick them as they crawl up into your body. You can also passively collect ticks from animals. And here you can see a photograph of mostly mammals being parasitized by ticks, fewer tick-amphibian associations, and a huge question mark on the bird, which is one of the examples that I'm going to show today in terms of research. So it is important to not only sample ticks in different type of habitats, but also understand, since ticks are really important vectors of bacteria, what are the bacteria composition of the same tick species in different habitats. This is work done by Helen Iser, a former PhD student at Stry, working with Patrick Jensen. And Helen came up with this great study that showed that adult ticks are quite specific in their relationship with their host. So one species tend to parasitize a group of closely phylogenetically related hosts. And this means that it's very unlikely that these ticks are going to transmit diseases outside that quite specific scenario. But we don't know much about the association between nymphs, immature stages of ticks, which could transmit pathogens as well between animals and humans. So today I'm going to be talking quickly about 3 molecular techniques: mass spectrometry, looking at protein. So the exocuticle of this insect have protein, and you can use that as a fingerprint to compare between species and within species and tell species apart. We're going to talk about the traditional DNA barcode, sequencing one sample for the cytochrome C oxidase 1 gene and comparing between species and telling them apart based on genetic distance. But here we're using a library of sequences that were collected from adult ticks. And then since we can't really identify morphologically the nymphs, we sequence the nymphs collected from birds and we match those sequences to the adult library. And then that's how we're gonna be able to identify the nymphs that are collected from birds. And at the end, we're gonna show data on metabarcoding, but the applicant here is the 16S, targeting specifically bacteria, not the DNA of the tick. And as I said, the bacteria composition can change under different quality of habitat, and that could mean that the tick could change the capacity to vector this bacteria as you collect it in forests as compared to degraded habitats. And so that's going to be the last study. Quickly, the workflow. So matrix assisted, this is a tricky one, laser distortion ionization mass spectrometry, time of flight. What you do basically is you use a small amount of tissue, you pull a leg from the tick, you macerate that leg with formic acid to extract the proteins, and then you mix that supernatant with a matrix. And what happens is that the sample co-crystallizes with the matrix. You sit it in a plate, you place it in this metal plate, and then you heat it with a laser. And so the molecules start gaining energy and start flying. So they fly through a vacuum tube towards a detector. And so the lighter molecules get detected faster, and the heavier molecules, the heavier ions get detected a little longer. And so what you end up having is this mass spectra, which represent the different ions, different peptides, different masses that are present embedded in the cuticle of the tick, in this particular case, the leg. And so when you compare spectra of individuals from the same species, they resemble visually quite similar each other, but when you compare it with a different species, then it's a different spectra. And so you go through quickly a whole filter processing steps to make sure that you're using good quality spectra. And then you come up with a library of average spectra for each of the species. And we did this with 18 species of ticks. And you can see here, there's phylogenetic signal in the spectra because spectra of different species within the same genus, Amblyoma, they're quite in the position of the picks and the number of picks, they're similarly and different from species that belong to a different genus, such as Ixodes and Hemophysalis. So visually, you can clearly see that there's some phylogenetic signal in this, but you can use then machine learning algorithms to classify the different samples. And this is a PCA, so it doesn't use information about the sample. It basically just clustered the samples based on their different features. And so you can quickly classify different genera as well as different species within the same genus. And so this is what they call unsupervised machine learning algorithms, meaning that we don't care about the pre-identification of the tick using morphology. Here we have least discriminant analysis. Here we pre-identify the tick and we train a model so that this predetermined groupings, and then with that information, it's able to assign an identification to the unknown samples. And through cross-validation, you can use this and quickly, at a 94% confidence, you can identify the species of ticks from an unknown sample. And so we have applied this to mosquitoes and sandflies as well. And there's a lot of advantages of using this tool. One that I will say is that we use ticks that were preserved in alcohol for the last 7 years, and we were still able to, the proteins don't degrade as fast as DNA. And so one of the disadvantages that I think it's important to mention is the cost of the machine. The machine costs $1.2 million. And so that's the, yeah, I know. But if you have it in your lab, or if you're collaborating with people that have that piece of equipment, then you could potentially use it. It's a lot cheaper than DNA amplification and sequencing in general. So the second technique that I want to showcase today is the traditional DNA barcode. And this is sequencing one individual with the 658 base pair that you guys were discussing earlier, and comparing DNA based on 2% cutoff of genetic distance. And so again, you can identify morphologically adult ticks, the ones at the far left, far right there, females and males, but you cannot do that with NIMS. And so what we did, we identified the 18 species of ticks that we have so far in Panama in our samples based on morphological characters, and we sequenced those and built up a library of DNA sequences. Then, and this is work done by Matthew Miller, former postdoc and PhD student as well as TRI. I collected birds across Panama. You can see all these 26 localities, and then you have samples from adult ticks as well as nymphs. So all the nymphs that were pulled from birds were sequenced and then compared to the DNA library from adults that were pre-identified morphologically. They worked on roughly 3,400 bird specimens from 26 locations, and this was really exciting. And so here's a phylogenetic tree. We have 18 clades, which represents the 18 species of adult ticks that we have in our initial library. And we were able to assign the 13 molecular clusters that we found in NIMS. 11 of them were confirmed, were identified taxonomically using DNA sequences. We have two barcode beans that we were not able to identify, one in the genus Ixodes and another one in the genus Hemophysalis. And we think that this could be either new reports in Panama or even new species. And so the cool thing about this is that NIMS also transmit diseases. They can do the same harm and damage as adult ticks. And so when you compare the estimate of richness and diversity between the traditional taxonomic tools such as morphology with more modern molecular tools such as DNA, you see that the estimates are quite similar. So both estimating between 13 and 14 species, the accumulation curve reaches a plateau quite quickly, and then it's very hard for you to find new species if you continue sampling. Another important aspect of this research that we found really interesting in is that certain ecological traits of the host, of the bird species, for example, tend to correlate with the probability of being infested with nymphs ticks. And so birds that are lowland birds that forage in the understory and their forest, the habitat that they use, are more likely to have nymph ticks as opposed to the other traits that I'm showing in the table. And so there's a higher risk for certain birds that are found in certain habitats. But when you look at the interaction between nymphs and birds, you can see here on your left the species of nymphs ticks. They're quite opportunistic. They feed upon a bunch of different birds that are not necessarily related phylogenetically or that share the same habitat. And so this is different from what we found with the adult ticks, which are really specific. And so this is something interesting in terms of disease transmission. Nymphs could be more catholic, more opportunistic, more generalist feeders, and that could increase the chances of getting diseases by nymph ticks. If you walk into a trail in BCI, for example, in the summer, that could be what is happening. So this study was also done to understand the role of birds in disease ecology. None of the ticks that vector pathogens to humans were found in birds. So based on this data, it seems like ticks don't play a role. Of course, we need to continue doing research on this, but at least initially, it doesn't seem like the vector Rocky Mountain spotted fever, which is the disease that we have found in certain cases in Panama, are parasitizing birds. And then the last technique that I want to quickly go over is the 16S ribosomal RNA amplicon metabarcoding technique. And again, this is not to look at the bulk sample of the environmental sample, but to look at the bacteria that are found in the tick. And so ticks are really good vectors of bacteria, and we want to know whether the tick diversity, the bacteria composition of bacteria diversity in that tick changes as the tick is collected in different type of habitats. So this is basically a combination of conventional PCR with multiplexing and next generation sequencing to generate thousands of reads from this amplicon, and then bioinformatically be able to identify the different species of bacteria found in the samples. People who know more than me about this, so I'm going to quickly skip this. So here's a accumulation curve again, and we not only sequence ticks, but we also sequence other vectors such as mosquitoes, sandflies, and Biden midges. And you can see the list of the species of hosts, the invertebrate hosts that we sequence samples from. And basically, individually, you can see that we reach a plateau real quick. And so it means that even if we keep sequencing these individuals, we won't be able to find more bacteria. In general, we found roughly 1,500 bacteria taxonomic unit or species. And the interesting aspect of this research is that when you classify the different bacteria found in all these species of insects, you see that there's also a phylogenetic signal in the bacteria, because all the species that belong to all the flies, mosquitoes, sandflies, and Biden midges, tend to have a more similar microbiome as compared to ticks, which are completely different. You see all the way to the corner, the ticks have a different composition of bacteria. And when you look at the proportion of bacteria, you also see that when you look at the flies, different species within each of these different 3 families of flies are quite similarly, not only in the bacteria composition, but in the proportion of all those bacteria as compared to ticks, which have a completely different group of bacteria found on ticks. And so when you compare, for example, 2 species of ticks belonging to the same genus, Amblyoma tapirellum and Amblyoma oblongutatum, you see that 40% of their bacteria associates are shared. But when you compare, for example, one of these 2 species to another species of tick that belong to a different genus, Haemophysalis justacochi, then you see that they barely share any bacterial associate. And so there's a group of unique bacteria of each of these species of ticks that allow you to identify them in an indicator analysis, and this can be done easily with all the species of ticks. And so you can use the association with bacteria to identify the ticks. And one of the important things that we saw as well is that when you look at flies, mosquitoes, sandflies collected in BCI that is not as degraded as chiotes and pavas, you see that the species composition of bacteria found in these insects changes across habitat with different qualities. And so in pavas, which is the most degraded sites that we had in our study, there's another group of bacteria, Rickettsia, and another group of bacteria that could potentially be pathogenic, could influence the vectorial capacity of these flies in degraded regions. And so we have seen this in various species of mosquitoes and sandflies. Same thing here. The proportion of certain bacteria increase as you go from BCI to places that have been degraded. We have more Wolbachia in pavas, for example, which is highly degraded. And so it seems like mosquitoes could potentially change their vector potential if they're found in different habitats. This is not the case of ticks. Ticks keep the same microbiome. Doesn't really matter where they're collected. So you see 3 species of ticks collected in BCI and also collected in Pavas, and you see sort of the same microbiome. So, and this may be because of the ectoparasitic habit, feeding on blood through the whole life cycle, that they just keep the same bacterial associate as compared to mosquitoes that develop in the water as larvae and then they emerge as adults. And so, again, and please correct me if I'm wrong, I think it's the same approach with bacteria. The metabarcoding approach is the only thing that changes is the molecular marker. Instead of 16S, you guys use CO1, right? So I guess in the future, we can apply this to bulk samples of arthropods, the vector diseases, and be able to also assess the diversity of this creature. As a general conclusion, I think it's important to understand those scientists that want to have a long-term research agenda to make sure, and this is, I'm talking about myself, I know you guys are more focused on community samples and environmental samples, but in my case, where we want to identify species and even describe new species, we try to use a small amount of tissue so that we can keep a voucher, a morphological voucher, and then perhaps use it later on. If another technique comes along, then we will be able to use that sample to confirm our initial identification. The metadata is also really important, geographic coordinates, so that you can use spatial analysis to understand range expansion, gene flow, all these other metrics that are relevant for vector diseases. And at the end, I think it is important as well for the students in general and the community that I've seen as a coordinator of the master program in entomology. I see this, the old guard and the new guard of taxonomies fighting over we should use CETA and size and color, and the newer scientists just want to use molecular tools. I really think that we need to integrate between these two schools and be able to extract the most information possibly from our efforts. And so this is going to help us to understand invasive species, cryptid speciation, and to more accurately assess community metrics such as species richness and species diversity. And with that, I would like to thank my group of students who have produced this data and have published this data, and also I invite you to check on our webpage. And I want to thank Owen and Christine and Norris, who have been really supportive from Stride to this effort. I also want to thank INDICASAT, the University of Panama, Luis Mejia, who's a great collaborator. Luis, thank you for all your support. And also folks at the Technological University of Panama, Fernando Marchan, Javier, and Rolando Gittens. And thank you for listening.
We'll have the question later.
Okay. Yeah.
So we'll have the question later for discussion. So we are now to switch to the next speaker, who is Jeremy Dewand, who was working at the University of Guelph, but now is working at the Smithsonian, at the National Museum of Natural History in the Smithsonian. Let's. Okay. We'll be ready in a few seconds. Et la présentation des SST.